Welcome everyone for our first regular business meeting of 2026. Happy New Year, and thank you all for joining us. I would like to call the meeting to order. Secretary Gary Havel, would you please read the minutes from the last regular meeting?
Yeah.
Thank you.
The 1256th regular meeting of the Entomological Society of Washington convened at 7 p.m. on the 7th of December 2025, gathering in room WG 33 of the National Museum of Natural History. Present were 13 members, 5 guests, and 30 virtual participants. At the request of Society President Talita Samoes, Recording Secretary Gary Havel read the minutes of the November meeting, and these were approved by the audience members. President Samoes summarized the events of 2025, noting that in January, Andrew Jansen resigned from his position as president. This unexpected situation led to Talita becoming society president. The annual banquet of the society was arranged by Talita, and the event was held at Busboys and Poets in early June. Some 50 members and guests attended the event to hear a career summary by Ted Schultz. The Young Entomologist group celebrated their 13th year, gaining new families. Editor Jerry Cook made remarkable progress in bringing the ESW proceedings issues up to date after the government shutdown. Membership and Communications Officer Matt Buffington conducted a membership drive that led to a current membership figure of 261 members. Program Chair Alan Norbaum oversaw a year that included speakers of various topics, including a presentation by 2 speakers from the Young Entomologist group. Webmaster Lourdes Chamorro initiated sales of merchandise from ESW and reported 7,900 website visitors. Curator Alyssa Seaman contended with a major overhaul for storage of papers and prepared material for removal by Mike Farrell of Clemson University. Next on the agenda of the evening was the annual election of society officers conducted by past President Don Weber, who handed out hard copies of the slate of candidates that were named last month. The candidates were elected by a unanimous vote and include the following: President Talita Samoes, President-elect Alan Norbaum, Recording Secretary Gary Havel, Membership and Communications Officer Matt Buffington, Treasurer Abigail Kula, Webmaster Lourdes Chamorro, Program Chair Renan Carreño, Editor Jerry Cook, and Curator Alyssa Seaman. Alan Norbaum mentioned that the proceedings special issue devoted to Wayne Mathis is expected very soon. Wayne was the Smithsonian curator for 35 years and is now residing in Utah. Virtually, Wayne gracefully thanked the Society for this notable honor. President Samoes noted that the Society website now has merchandise for sale and passed around a mug that is now available on the site. Program Chair Alan Norbaum introduced the speaker of the evening, Rodrigo Diaz from Sao Paulo University in Brazil, who has for the past year been a postdoctoral fellow with the Smithsonian Institution. The title of the talk was The Most Diverse Parasitoid Flies, Tachynidae, Diversity, and Evolution. Tachynidae may be the largest family of flies. Currently, they are known as parasites of 7 orders of insects. They can be utilized for biological control and integrated pest management. They have been used in control of Japanese beetles. There are 8,600 known species, and this figure will be higher in the future. They are most diverse in the neotropics. There are 5 subfamilies. Knowledge of taxonomy and biology is not well known. Oviposition is external and ovipositors are diverse. Through the use of many slides, the diversity of these flies was demonstrated, including images of remarkable mimicry of wasp and other flies. Townsend was a prolific worker in tachynids, but his studies were flawed. At the Smithsonian, there are some 1,000 types of taquinas. Future studies should include research on species boundaries. That's what I had.
Thank you, Gary.
Sure.
Does anybody has an additional correction?
Did I hear that the Kennedy had how many species?
I had down 8,600. I don't know.
Oh, okay.
Yeah.
I misheard. I thought you said
Okay.
86 million.
Yeah.
Any other correction? Okay. Can I have a motion for the minutes to be approved as read? Second. All in favor? Thank you. So the minutes is approved. We move now to officer reports. Does any officer here or on Zoom has a report for us tonight?
Good evening, folks, and welcome to the first meeting of 2026. Okay, I am Matt Buffington, the membership secretary. More on that later. We have several new people that have joined the society since the last regular meeting. These include the following individuals: Carson Kane of Washington, D.C., interested in conservation, climate change, and Kalimbala. Dan Frischtak of Potomac, Maryland. No particular interest of subject, but that's okay. John Hash, who I do know from the Harity Lab of Loma Linda, California. His interest is in Diptera systematics. David Allred of Sterling, Virginia. No listed interest. Here's an interesting one. Steve Gamari. Steve Gamari was part of my graduate program in a loose way. A fellow grad student of the P program says he let his membership lapse, so he's considering himself a new member, interested in all things Diptera. Vince Brock of Tyler, Texas, interested in Hymenoptera, and Zach Leahy, who is listed as Charleston, South Carolina, but I know he's actually in Gainesville, Florida. He's a new scale systematist there with the FSCA. So those are all new members to be read in to the roster to become full members at the next meeting. Thank you very much.
Thank you very much.
Oh, look at that.
Curator Seaman.
I just have one short announcement. I'm sure if you're watching on Zoom, you've noticed we're going through some technical upgrades. So we have a new microphone. It should be a lot easier and more stable on Zoom to hear what we're all going through. So let us know if there are any issues with that. That's all there.
Thank you very much. Any other officer report? Do we have anything on Zoom? No? Thank you. So we move now for the introduction of new members or visitors. So I would like to open the floor for anybody who is here for the first time and would like to introduce themselves. Thank you and welcome.
I was an ICCN commissioner for almost 8 years, but that ended last.
Thank you very much and welcome. I had a chance to watch Madeline working over summer, and I'm very happy to hear the news. Anyone else? If anybody on Zoom would like to introduce themselves, please raise your hands and we're gonna get you unmuted. So let's move to unfinished business. Does anybody have a unfinished business from previous meetings that you'd like to adjourn now, to address now? No? New business, I have one. I'd like to introduce to you our newly appointed officer. By the authority of the president, I'm pleased to formally appoint Cecilia Escobar as our social media secretary. Cecilia has been acting in this role for some time, and we are happy to now make it official. So welcome, Cici.
Follow our Instagram if you haven't yet. She's done amazing with it. And YouTube.
Yes. And probably for the next year, we're gonna just have it as an official office on the bylaws. And then we're gonna split membership and communication with membership and communication and social media. Okay, any other new business? Okay, do you?
Just a quick update. Lita, CC, Dave Furr, and Alyssa and I have been working hard to clear out all of our backlogs of procedures of Entomological, Washington, to remove them from the attic, remove them from the building, to Mike Farrow, who is going to be keeping them alive and well, Clemson. We've got everything out of the attic. We're now going to be moving on to the Entomological Annex, which we have a small room in the library cleanup, plus part of the basement of the library. But we are making a lot of progress. We're happy. It's started during COVID, I think, and it's finally beginning. Like things are beginning.
Yeah. Thank you all.
Correction, you didn't get out everything because got to 6,500 pounds and his truck was only 6,100. By the time he got to Southern Virginia, he had like
Oh.
You got at least 4 trips.
we still have 4 pallets and a couple cards of
Okay, so we will move on presentations of notes and exhibition of specimens. And I also have one thing today. This is the gavel box, and Matt just found the beetle that made this. So if you could pass along carefully. And if anybody has something else, you could come up here and introduce and then pass as well.
I do, but I'm going to do it from over here.
Okay.
So when I was cleaning out David Amson's house recently, I found some interesting illustrations. Oh. This one done by Bill McGowan, and it's a caricature in a sense of C.B. Riley. Coming out of an acorn because he described a moth, I believe it was. It was acorn moth from Massachusetts.
Wow.
So it's a bit of a caricature. Kind of knew him. Knows him.
Where's that ending up, Dave?
We don't know yet.
They got a picture of it.
Yeah. Found some other
Just
ones.
leave it over there. I'll get it later.
And also, I found by the same artist, Joe McCowan. Oh, sure.
Yes.
I also found a couple of illustrations or caricatures, if you want to call them that. Oh, Adamski by the same illustrator. This one. This one.
That was spot on.
Finally, something really very rare. Two items, actually. This is the trumpet leaf miner. Original color illustration done in 1920 by Robert Snodgrass. If any of you know much about insect morphology, he was the guru. His book on insect morphology in 1935 is what I use when I took insect morphology.org. This is an original done by Snodgrass. and it's amazing. But I would ask you not to touch the front of it at all. There's one more. There were several, but I brought two. This one was also by Snodgrass, serpentine leaf miner of apple. Also in 1920. When you look at these, you'll be amazed at the detail, especially the results. It's the whole life cycle. There were others too. Nice. Snodgrass. Speaking of Cornell, was a guy, a few Cornellians in the room. Some people know about Tom Eisner. And when I was at the MCZ at Harvard, I was walking through the halls with Ed Wilson and Tom Eisner. And Eisner told me, he said, one of the best experiences of my life was after I finished at Harvard. He was a PhD student, Frank Carpenter at Harvard. He came down here to Washington, spent 2 days with Snodgrass, and said that was one of the most important things. It's like, most of you don't know probably, that Snodgrass was hired at Cornell to teach insect morphology. He eventually became a chemical ecologist. So most of them. So,
Thank you.
yeah.
I heard that about an entomologist who originated from this area, from Falls Church, and she's now doing a program for knitting entomological creatures. And the one that I went to a presentation that she gave at a yarn store in Falls Church called Entangled, as part of her series called Local Critters for Local Knitters, and her name is Meredith Wilmot. She's currently a PhD student at the University of Arizona in Phoenix. So the critter she chose for Falls Church, Virginia, was the six-spot tiger beetle. So this is the six-spotted tiger beetle cowl pattern. It is free to anyone who visits Entangled Yarn in Falls Church. And it's a knitting store. You can get this when you go in and get your knitting supplies and make it yourself. I have not made this one yet, but I have done another one of her designs. This is the collection's cowl. It includes lots of different insects, Diptera. There's a lacewing up here, katydid, Luna moth, cicada, and leaf insect. Oh, and this one's a monarch caterpillar, the long one. And then this is an earwig. I think I got them all now. Yeah, so lots and lots of different insects you can knit. And if you look up her name, Meredith Wilmot, you can find all of the knitting patterns she's made and find out more about her research. She is, I think, specializes in leaf hoppers and plant hoppers. And she said she did used to go to ESW in like 2018 or 2019, but I never met her personally when she was going. But maybe some of you have. All right.
Thank you very much.
Still some picture time.
Oh, we got a great show and tell today. Do we have something else?
Just one quick note, not a show and tell. Many of you are members of the Entomological Society of America, and they are currently holding a vote on changing their bylaws. I was very involved with the NSOC of Washington change in bylaws and much favored it. I do not favor the current proposed changes to the bylaws of the ESA, and I don't want to use up ESW time with that, but if you have questions about that, see me afterwards.
Thank you. Anything else? Okay, so we move now to the presentation of the announced topic for the evening. Ted Schultz is Katherine's supervisor. Ted, can you introduce her to us?
Good evening, everyone. It's my pleasure to introduce the speaker for this evening, Katherine Malinski. Katherine got her PhD in 2024 from the University of North Carolina, Chapel Hill, where she worked with Joel Kingsolver on parasitoid host symbiosis. She then immediately came here and as a Peter Buck postdoctoral fellow, where she's working in my lab. She's working on an aspect of fungus farming ant biology. The title of her postdoc project is Microbial Coevolution in Fungus Farming Ant Gardens, How Microbial Communities Vary Across Ant Species, Nests, and Habitats. And that's correct, right? That's, yeah. And what this involves is going through lots and lots and lots of fungus gardens that we've preserved over the years, and doing environmental DNA, what's it called, metabarcoding, looking specifically for bacteria, fungi, arthropods, and trying to characterize what the communities and the stable members of the communities and maybe the not so stable members over vast geographic areas. So, but, and I believe that will overlap with what she's going to talk about this evening. So, yeah.
Thank you, Ted. So hi, everyone. As Ted mentioned, my name is Katherine Malinski, and I am a Peter Buck Postdoctoral Research Fellow, where I have been working with Ted Schultz and Jeffrey Sosa Calvo for the last about a year and a half now on fungus farming ants. And I am really an ecologist at heart, but I tried to put a lot of insects and a little bit of systematics into this talk for all of you. So you're welcome for that. So I'm going to tell you a little bit about what I've learned over the last year and a half from working in this system, and then also some of the results from this postdoctoral research that we have. Are they okay on Zoom? Yeah. Okay, cool. All right. So just to go ahead and get started, I'll play this video for you so you have something to look at while I ramble. But if you take nothing else away from this talk, the sort of main thing that I want to get across with these slides is that symbioses are just really fascinating and have fascinated scientists for a very long time. But being able to study symbioses within a community context where you've got not just two interacting species, but multiple interacting species is a really rare opportunity. And it's something that the fungus farming ants have allowed us to do. And so that's what I'll be focusing on for this talk, both on small and large scales. Okay, so who are the fungus farming ants? So as their name suggests, they are ants. They are farmers and they farm fungi. So they carry substrate back to their colonies where they don't eat the substrate, but they instead feed it to their gardens and sort of fertilize their gardens with this. In this case, the case of leafcutters, leaves, but they'll also forage other material for their gardens as well. So they're doing agriculture. It's just like we humans do agriculture, but it's ants growing fungi. And so, the ants have been doing this for over 60 million years, whereas we've only been doing this for about 10,000 years. So there's really a lot that we can learn from these ancient agriculturalists. So just a few key background pieces. This is a monophyletic group of 250 described species in the tribe Atini. So you will hear me refer to these ants as Atine ants. They're exclusively New World, so they're only found in the Western Hemisphere, primarily Central and South America, but we do have some North American species as well. And they evolved around 66 million years ago, which is right around the time that another really big event happened, the KPG extinction event.
And so this is one of my favorite parts of fungus farming ant evolutionary history. So if you look at the fungus farming ant phylogeny, you see that the characteristic of fungus farming evolved right around the time that the dinosaurs went extinct. And the sort of story here is that in this new environment, you had a lot of dead and decaying material. And so having an association with an organism that can break down that material would have been really advantageous. And so that's not really what I study, but that's just a piece of their evolution that I think is just really, really cool. Okay, so fast forward 66 million years, and now we have these 250 described species that farm fungus. And they do this by carrying foraged material back to their gardens. So this is an image of what one of those typical gardens kind of looks like. But they sort of eat different things, and they'll feed their gardens different things depending on the fungi that they farm, which is largely dependent on the mode or system of agriculture that they practice. And so I just want to go through the very basics of these different systems. So 66 million years is a long time, and they had a lot of time to diversify the way that they grow fungus and also the fungi that they grow. So there's 4 systems. We're going to start with the first one, which is lower agriculture. So this is a video of a lower atine ant garden. It's about the size of a baseball. So this is the most common type of fungus farming. It's also the most basal form. So about 80% of the phylogeny practices this form of agriculture. And the fungal cultivars are actually facultative symbionts. So they can grow on their own. Whereas ants are obligate symbionts, so they can't survive without this fungus. As I mentioned, they're typically small, usually only 1 garden chamber. They're really easy to miss. I was really bad at finding these in the field. Jeffrey had to very frequently point them out to me to dig up while he went and found some more on his own. So that's lower agriculture. I like to think of these as like little garden parties, just sort of happening out in the forest. So from lower agriculture, you get some diversification into other more advanced or specialized systems of agriculture, including yeast agriculture. So this is a form of agriculture that's practiced by a species group of about 20 species. And what's characteristic about this form of agriculture is they grow their fungal cultivars in this yeast phase, where instead of that hyphal, mycelial, like spongy, cottony fungus, they grow in these tiny little balls. This is the only case of this type of growth in this family of fungus. So it's pretty interesting and pretty weird that they do this. And it's thought that one of the reasons that they do this is potentially to prevent the spread of a really prominent crop pest. But we'll get into that a little bit more later. So that's yeast agriculture. Next up, we have coral fungus agriculture, which is another weirdo. So this is a system of agriculture practiced by ants in the genus Apterostigma. And what they do is they grow fungi in the family Pteriolaceae, which is the coral fungus. And this is a sort of distantly related type of fungus. So what this basically means is that at some point in their evolutionary history, they made a switch to growing something new. So this would be like if your whole family grew soybeans and you suddenly decided to switch to corn, for example. So that is the coral fungus growers. And coral fungi are kind of weird. So this is one particular species of coral fungus where the hyphae will grow into these really long aerial hyphal, I don't know, not roots, just aerial hyphae basically. And the ants will stitch them together to form a tent-like structure over their gardens and basically let them grow in these more exposed areas. So that's coral fungus. And then last but not least, we have the higher agricultural system, which is practiced by the leaf cutters, as well as some other ants as well. So what's special about higher agriculture is that these ants are actually growing a domesticated fungal cultivar, and it's an obligate symbiont with the ants. So the fungus can't grow without the ants. And in all of these cases, the ants can't grow without the fungus. This fungus is polyploid, it's multinucleated, and it produces these specialized structures called gongolidia, which are basically swollen hyphal tips on the fungus that the ants can then eat. They're really nutritious. You can think of them kind of like little lollipop rewards for growing this fungus. And it enables these colonies to become really complex and really large. So sometimes even the size of like small city blocks. And so you can imagine that sustaining a colony of this size requires a lot of plant material. So these guys are voracious herbivores. They are major crop and forest pests in the areas that they occur. Okay, so those are the 4 forms or systems of agriculture. And the sort of central thread of all of this is the tight coevolution that occurs between the fungus and the ants. And so to illustrate this, we have a recent co-phylogeny, dated co-phylogeny of the fungi on the left and the ants on the right. This was produced by Ted and Jeffrey and colleagues really not that long ago. And what's cool about this is it's the first really well-supported fungal phylogeny that we have. And so it's cleared up a lot of the story of ant and fungal coevolution. And so what you're seeing here in the different colors are those different forms of agriculture. So pink is coral fungus, light yellow is the lower fungus, lower agriculture fungus, green being the yeast fungus agriculture, and then blue and then that dark yellow being higher attine fungus farming, with yellow being the leaf cutting ants. And the basic gist here is that there's really tight coevolution going on between the fungi and the ants, which is illustrated by the mostly parallel lines going on between the blocks of the sort of same colors. And so this coevolutionary story is really why fungus farming ants have become such a textbook example of coevolutionary symbioses. So that, I think, is most of the background. And so, you know, the fact that this is a textbook example of symbioses has really led to the study of other symbionts that exist in these gardens. And so we can kind of use this system to study ecology and coevolution from a community context, as I've said before. So I'm going to go over what some of those symbionts that occur in these gardens look like and what they do. And just to give you an idea of the sort of levels of biodiversity that we're talking about in these colonies. So there have been cases of excavations of single colonies, in this case, leafcutter ants. And these researchers recorded every species that they saw. So in the first case, they found just a multitude of species of worms, nematodes, pseudoscorpions, other invertebrates. And in the case of the Ada Sexton's garden that was dug up, they found a whole bunch of beetles, bugs, mollusks, flies, even reptiles in some cases. And so these environments can form really cool niches for other species to exploit. So we're going to get into that. First and foremost, different species will specialize on different parts of these fungus farming ant gardens, right? Because they can take up entire city blocks. There's quite a lot of space. And so I just want to draw your attention to sort of 2 areas in this fungus garden. This is a leafcutter ant garden. The first of these being the garden chambers that you see sort of pockmarking the soil here. So each of those will contain a piece of that spongy fungus garden. And then the second is these dump chambers. So fungus farming ants are super hygienic and they will weed their garden to basically remove anything that they don't like, and they'll just dump them into these dump chambers. And so you can imagine there's a lot of detritivores and sort of other species that can really enjoy being in those dump chambers. So moving forward with the talk, I'm just going to use this little cartoony, simplified illustration of a fungus farming ant garden. with just one fungal chamber and one waste chamber to really illustrate these sort of microclimates that the symbionts can live in. With the addition that there's one other environment that symbionts can live in, and that is the reproductive phase of these ants. So in leafcutter ants, when they are ready to go off on their own and start their own colonies, queens will grab a piece of their natal fungus garden in their mouths, which we call the infrabuccal pellet. They'll then fly off away from the garden and go seed their garden somewhere else. And so you can imagine that there's a lot of other species that use this as a mode of dispersal. Okay, so going over what the rest of this talk is going to look like, we are going to talk about all sorts of symbionts in these different agricultural systems, and we're going to go from big to small. So first we're going to overview vertebrates, and then we'll talk about invertebrates, where I'll spend a lot of time because this is the Entomological Society of Washington after all. Then we're going to move on to more of my specialty, which is microbial symbionts, but I promise it'll be cool. And then finally, we're going to go over some of the results from what Ted mentioned as this metabarcoding project that I've undertaken here at the museum, which is really going through the fungus gardens that we have housed here and trying to learn everything that we can learn about the communities that are inside of them. Okay, so starting off with vertebrates. So within vertebrates, there are quite a few examples of egg-laying vertebrates that use fungus gardens for incubation. And so it's thought that they do this because this is a cavity that already exists. It's easy to get into. It's well defended if you can convince the ants that your eggs are supposed to be there. And these fungus gardens are really an environmentally buffered condition. So in some cases, they can be an ideal temperature for incubation. So the cases where they have found eggs in these gardens, a lot of the times they'll be completely encased in the fungus, kind of like a little pillow. But of course, you have to be able to evade recognition by the ants or convince the ants that your eggs are supposed to be there in the first place. And so this has led to a lot of really cool adaptations for essentially mimicry or disguising yourself. So in the case of this frog up in the top right corner, this frog has cuticular hydrocarbons that essentially disguise it from the ants. And enables it to go deep down into atta gardens where it can then lay its eggs. So there's also a lot of squamates that have been reported to lay eggs in fungus-farming ant gardens, specifically leafcutter gardens. So 20 species have been reported to date. Several of these have been reported to follow ant foraging trails, including one worm lizard that was recorded to follow an ant forage trail for over 50 meters, which is kind of a long way. And this suggests that they have some sort of chemosensory adaptation to be able to do this, but it's really not well studied the extent to which these relationships are obligate versus facultative. We just kind of don't know a whole lot about them. It's been mostly observational studies. And something that I found really interesting was that there's not a whole lot of reports of these squamates actually consuming the ants or the fungus. That certainly does happen. But in a lot of these cases, the researchers dissected out the guts or the fecal matter of these squamates and found that they were not consuming the ants or the fungus, which I thought was surprising. So that was mostly higher agriculture and mostly leafcutter ant nests, which again are really big. You can imagine there's a lot of space for a little snake to be squirming around in. But what about lower agriculture? So these are smaller nests, right? There's not a whole lot of space, but that really does not stop them. So there is at least one known case of snake oviposition into a lower attine nest. I thought this study was really interesting because the researchers essentially found that there was a blind snake that had oviposited into an apterostigma nest, which is a lower attine agricultural nest. And they observed these ants actually covering the eggs with their fungus garden and antenating the eggs. So essentially caring for the eggs like they would their own brood. And they also, in this study, implanted a fake egg. It was basically a sterilized piece of plastic egg to try to see if this was just a response to a foreign body. And the ants just ignored that fake egg completely. So it seems like these eggs have some sort of mechanism by which they're convincing the ants that they mean them no harm. And in this case, the snakes did not eat the ants or the fungus garden as well, which is similar to what we saw in the higher attines. Okay, so that's all we're going to say about vertebrates. It's cool, but vertebrates. So now we're going to move on to invertebrates, which we have quite a few cases of symbioses with fungus farming ants. And we're going to start with our hitchhiker taxa. So within the arachnids, there are quite a few notable hitchhikers. There are at least 7 families of spiders that have been observed in fungus farming ant gardens. Most of these have been observed hunting in waste deposits or empty chambers, so finding workers of the nest that are passing by, or other invertebrates that are in those waste piles. But there's the case of one genus of spider known as Atticobius that is apparently adapted to living with leafcutter ants and will find leafcutter ants and grab onto them like a backpack and then ride them into the fungus gardens where they then are presumed to eat fungus and brood. And we have a video of this. If you look closely, I know it's a little bit small. You can see a spider that is too big to have any business going after an ant that size, riding the ant like a backpack into the fungus garden. So that's Atticobius, rides workers into nests like a backpack. And it's thought that they hide from the ants by stealing cuticular hydrocarbons. So as you'll see in this next video, these spiders will rub their legs up against the ants in an apparent attempt to disguise themselves. So you can see it's sort of rubbing its legs against the ants and harassing the ants. All right. So that's Atticobius. It's been found that these are obligate symbionts. So in the lab, when you take the ants away from the spiders, they die. And one other really cool thing about these spiders is that they have this really interesting dispersal mechanism where when it's time to leave the garden, they will grab onto an alate and ride them out of the nest. But they've never been found in incipient gardens. And so it's thought that they're detaching mid-flight or after the ants land, and then finding mature colonies to disperse to, which makes sense, right? There's no garden or brood to eat in an incipient garden. So yeah, that's spiders. Moving on, oh yeah, we have our little cartoon spider on the queen there. Moving on to mites. So there are quite a few mites that have important interactions with ants. In some cases, mites have been demonstrated to cover the head and limbs of ants, restricting their movements in some cases. And it's thought that they will ride ants traversing to other areas of the colony. There's our mite. All right, our next hitchhiker, some cockroaches. So cockroaches in the genus Attaphylla are duly named because they have an obligate parasitic relationship with fungus farming ants. This is what they look like. They are really cute. And they have a sort of similar life history to the spiders in that they will ride alates, but only female alates in this case, to disperse to mature colonies. So again, this is an image of one of the cockroaches on a queen. Again, they're not sticking around with the queen as she's forming her new colony, which makes sense. But these cockroaches are apparently adapted for life with ants. So they have modified tarsal pads that allow them to grab onto ant cuticles better, which is probably important if you're flying with them. And then they also have modified maxillary palps that presumably can help them locate ant forage trails after they have dispersed. Okay, moving on to Hymenoptera. So there's quite a few interesting Hymenoptera that associate with fungus farming ants. The first of which having to be parasitoids, because of course. These are diapriid wasps. There are a couple genera that have been found in fungus farming ant gardens. In some cases, they can parasitize up to 70% of the colonies found in the field, which is a strong selective pressure. And so here you can see they're parasitoids of ant pupae. And so you can see these are some developing diapriid wasps. And what's interesting is that they're undetected in ant colonies as larvae. So the ants will care for them the same way they'll care for their own brood. But once they eclose, the ants quickly recognize them and will attack them if they're not quick enough to get out of the fungus garden. So it seems like that sort of masking or disguising themselves does not last once they eclose as adults. There's our parasitoid. Okay. So next up in the Hymenoptera, we have raiders. These are agropredators, with the best known case being Namtogenes. These are ants that essentially come into the fungus garden, overwhelm the workers, and sort of boot them out, and then really just usurp the colony. So they'll eat fungus, they'll eat brood, they'll decimate the colony, and they don't take care of the fungus garden. They use up the resources and then they leave. So there's our Namtogenes raiders. And then we have the case of social parasites, which are really interesting because they will come into fungus farming ant gardens and sort of establish long-term relationships with them. So the best known example of this being megalomyrmex. And megalomyrmex are interesting because they sort of exist on this spectrum of really, really parasitic to commensal to sometimes mutualistic. So there's some species of megalomyrmex that kind of function more as raiders. They'll come in and just eat all of the fungus and the brood. But then there's other species like this one shown here, which you can just tell from its face. This is a good guy. And so they will come in and they'll sort of live alongside the fungus farming ants. They are consuming, fungus garden and resources, so they are, technically parasitic. But what's interesting about these guys is that when another raider comes in, like Namtogenes or something like that, they will actually come to the aid of the fungus garden, and they have a really potent alkaloid venom. And so it's been shown that when a fungus farming ant colony has this species of megalomyrmex and then gets raided, they have a better success of fighting off those raiders relative to if they did not have this social parasite. So it's this really interesting trade-off between you are being parasitized, but you have some protection against some other species of raiders and things like that. There's our little social parasite. Okay, so we're gonna talk about Diptera for all my fly people. So there are some fly parasitoids of fungus farming ants, with the best studied example being Atopheles, I don't quite know how to say it. But these guys have been studied pretty extensively for their ability to control leafcutter ant populations because they're pest and forest species, pests of crops and forests. And so fungus farming ants have developed a sort of defense strategy against these parasitoids where a minima worker will ride on a piece of the substrate. This is a cartoon because I couldn't find a good picture of it, but this is kind of what this looks like. And they will, like, sort of from above be able to fight off the Diptera parasitoids. So that's sort of a cool parasitoid evasion strategy that's been developed. And then we have the sort of more ephemeral members of the fungus garden communities. So this includes springtails, silverfish, crickets have been demonstrated to exist in these gardens, as well as beetles and some Lepidoptera that have been carried into the gardens from forage substrate where they then exist for a period of time. And so these species exist and feed on waste deposits and or fungus gardens. And there's one sort of interesting interaction where silverfish will actually groom fungus farming ants, like tickle their little faces, and sort of stimulate trophallaxis, where the ants will regurgitate whatever they had in their guts for the silverfish to eat. So that's just kind of a weird, cool little aside. Then there are the species that specialize on the trash piles. As you can imagine, this includes a lot of annelids, nematodes. There are some bugs and beetles that will hunt other members of the trash lovers. And the nematodes are common in waste, but they can also serve as gut parasites of leafcutter ants as well. And as I mentioned, the trash is a great hunting ground. So there are spiders and beetles and bugs that will hunt other invertebrates that live in these waste piles. So these are kind of cool little ecosystems in and of themselves. Okay, I hope that was enough invertebrate stuff for you all. We're gonna move on to microbes now. And what's really interesting about some of the microbes that we've seen in this system is that it really adds to the coevolutionary story between the fungi and the ants that have co-evolved with each other. And so one really important microbial parasite in these gardens is Escovopsis. And Escovopsis is a fungal parasite that causes crop disease. So this is basically a crop disease, if you will. And it competes with the ants to consume the fungus garden. So this is an image of a healthy garden, and then here we have an image of a diseased garden. So Escovopsis fungal parasites will form these sort of brown and yellow and pinkish lesions on the garden where they're infecting it. And this Escovopsis parasite has never been found anywhere other than inside of fungus farming ant gardens. So it's thought to have a really close evolutionary relationship with fungus farming ants. And we do see support for that when we look at the cophylogeny between ants and Escovopsis. So what we're sort of seeing here is that on a sort of broad scale, clades of Escovopsis have co-evolved with clades of fungus farming ants. And so we do see evidence for some coevolution between Escovopsis and the ants. And what you may or may not notice here is that there is no yeast agriculture on this Escovopsis list of Escovopsis targets. And so that's because we've never found Escovopsis in yeast gardens. So as I sort of mentioned before, it's thought that yeast agriculture might be a way to avoid this sort of really important crop pest. So that's one way of dealing with a disease, just grow a crop that is resistant altogether. But another way to deal with garden diseases is through basically biopesticides. So the ants can control Escovopsis as well as other garden diseases with this bacteria that they cultivate known as actinobacteria. And so this includes bacteria that they grow on integumental crypts that produces secretions that actually simulates the growth of this actinobacteria. And this actinobacteria is antifungal to Escovopsis. So here you're seeing this brown stuff is Escovopsis. In this top panel, we have the growth of a Streptomyces, which is an actinobacteria, from a leafcutter nest, and you can see this sort of zone of inhibition. And then this is a control bacteria that is not an actinobacteria. So they do produce antibiotics, and they're used by the ants sort of as a biopesticide. And so what this system really tells us is that it's not just this one-to-one coevolution between the fungal cultivar and the fungus farming ants. But we also know that there is this crop pest that has important negative consequences on the fungal cultivar. And this, fungus farming ant will actively cultivate actinobacteria, which it uses as a biocontrol mechanism for that crop pest. And so this just really sort of expands our understanding of some of the ecological and coevolutionary dynamics that are happening in these fungus farming ant gardens. And it's really paved the way for us to think about other sort of microbes and other symbionts that might be playing a role in what's going on in these colonies. So for example, we've also found that actinobacteria can provide benefits to preventing disease from an important entomopathogen called metarhizium, which the ants will get when they're foraging out in the environment. We also know that it can provide protection against opportunist crop pests like endophytic fungi that come into the garden from foraged material. So it seems like actinobacteria, instead of just being specific to Escovopsis, might provide a whole suite of benefits for the ants and the garden. And then thinking about other bacteria that play a role in these gardens. So there are some microbes that are involved in defense. So proteobacteria in the genus Burkholderia will selectively inhibit Escovopsis, but not the fungal cultivar. And then we also know that lactobacillus species are used to combat opportunistic crop pest endophytic fungi. So that's defense. There's also a lot of really cool nutritional and metabolic microbes that exist in these gardens. So one of these being mollicutes, which are gut bacteria, and the ants use mollicutes basically to fertilize their gardens. So nitrogen is being fixed in the gut, and then they use their fecal droplets to sort of promote growth in the fungal cultivar. And then we also know that there are several species of yeast that detoxify plant secondary metabolites that are brought into the garden from foraged plant material. And so those also exist in the gardens as well, essentially providing benefits to the ant in the garden. I don't know if I want to talk about this right now. I'll just sort of mention this briefly. This was a really cool comparative study that was done comparing the bacteria that associate with different insects that practice fungiculture. And so there's a lot going on, but basically what they did was they compared pine beetles, ambrosia beetles, and fungus-growing ants, and they looked at the composition of their gut microbes. And they found basically there's this story of convergent evolution going on within their microbiome. So essentially, if you're practicing fungiculture, you're probably going to have a microbiome that is dominated by gamma proteobacteria. I think that's all I want to say about that. I could go on, but I won't. So that is just sort of an overview of some of the microbial symbionts that exist in these gardens and their roles. And so now I want to pivot to sort of what we've learned so far from metabarcoding fungus farming ant garden samples here at the museum. And the sort of goal of this work was really to sort of characterize what the community looks like within fungus gardens with regard to bacteria, fungi, and then also insects, because pretty much all we know is from leafcutter ants. And so we sort of wanted to take a comprehensive approach to answering this question. And I will just say that this work was really only possible because of all of the people who came before me doing all of these collections, which you'll see on the next slide is a bit of metadata of what some of our collection looks like. So as you can see, we have fungus gardens from across the fungus farming ant range. There are some more to be added to this from some recent collections. I didn't do any of these were all other people. And so we're sort of looking at the gardens taking a biogeographic approach, and then also taking a phylogenetic approach to sort of trying to understand how these microbial communities mediate or are mediated by the ant and fungal phylogeny. So this work is currently in progress. And so what I want to show you now is just one of the sort of little studies that we ran as a part of this, which was really focused on characterizing the bacterial microbiomes of lower atine fungus farming ants on a sort of local level. So here's a video of a lower atine garden. Again, they're small, they're inconspicuous. We know a lot, not a lot. We know some about how the bacterial microbiomes within the gardens are structured from leafcutter ants, and we know almost nothing about how they're structured in lower atine ants. And so that's what I wanted to characterize with this study. So to do this, we did what I like to call the riverbank spatial survey. And this was from recent fieldwork that was done down in Panama. And what we really wanted to know was, what sort of role does the environment play in internal garden community structure? Are the ants actively structuring their microbial communities inside the gardens, or is it just like whatever's in the environment? Is sort of the basic gist. So we traveled down to Gamboa, Panama. We found it's called, I'm calling it riverbank, but it's really more of a creek, where we know that there are a lot of lower atine ant gardens. And we essentially walked along this 200 meters of riverbank, sampling every lower fungus farming ant garden that we could find. And what we did was we took samples of the internal garden. So this is what some of this work looked like. You can see here, this is a piece, or maybe an entire fungus farming ant garden. So we sampled inside of the gardens, and then we also sampled 10 centimeters adjacent to the nest opening in the soil. And so that really gives us a metric of, okay, what microbes are in the environment at this place? And then what microbes are in the environment in the fungus garden at this place? Again, with the goal of answering the question, does the environment or does the ant and fungal host species play a bigger role in structuring garden communities? Because if there's no structure by ant species, you would expect that all of the species along this riverbank, it's really just the location along the riverbank that determines what is in your garden. So we tested this explicitly. And so now I'm going to show you some of the data. What we're showing first are going to be metabarcoding results from our fungal marker, which was ITS2. And so what this is showing is a principal coordinate analysis plot of the data, which in this case is alternate sequence variance. You can just think of it as community structure. So the axes here show the amount of variation in the data. The points here, each individual point is an individual garden, and the colors are different species of fungus farming ant. And I should mention the X's here are the means of those points. And what I want to sort of draw your attention to first is this. This is the results for inside of the garden. And then on the right, we have results for the external soil community. But this plot I'm using as a sort of positive control for interpretation, because I didn't remove any of the cultivar sequences. So this includes fungal cultivar in this plot. And of course, we know that ants play a really strong role in structuring the communities of their fungal cultivars because of that one-to-one coevolution. So this is what we expect to see if the ant host has a strong and important role in structuring the community. And we do see this supported by our statistical test, Mantel test in this case, So host species was a significant predictor of community composition, but space was only marginally significant, which again, this is what we expected, given that this is full of just basically cultivar sequences. So you can see that, the points mostly cluster. There's not really a whole lot of overlap going on. And there's weak non-significant influence of space on the sequences found within the gardens. But now if we look at the soil community structure, so the fungus that's found in the soil just adjacent to the nest, we can see that that relationship mostly goes away. So there's a lot of overlap between those points. Statistically speaking, we didn't see anything falling out as being a significant predictor of community structure. So within the soil, there's no evidence of a host signature, maybe some weak evidence for a spatial signature. But again, keep in mind that for the garden, this is not indicative of a microbiome. This is more indicative of the fungal cultivars themselves. Okay, so that's the fungus. So now I want to show you our results for the bacterial markers, which we used 16S for. And this was really kind of what we were going for with this study. So these are the results for the internal garden community of bacteria. And so you can see there's some clustering, but it really overlaps quite a bit. And statistically speaking, we didn't see a significant effect of space or host species. So there's really no differences between the bacterial microbiomes of these different fungus-farming ant species within their gardens. And also really no statistical difference depending on where you're at in the riverbank. Yeah, I just said that. But then if you move to the external soil community, we actually did see a strong effect of space on the community structure of the soil adjacent to the nests. So in other words, once you move outside of the fungus farming ant garden, where you are along this riverbank is what determines your microbial community. And so what this really kind of tells us is that there are different mechanisms that are structuring the bacterial communities within fungus gardens relative to the environment. So it's not just whatever's in the environment, right? There's some sort of filtering going on in these fungus gardens. So again, we find that bacterial microbiomes in gardens and soil differ. And the fact that, there wasn't a difference between species when we looked at bacterial community composition within the gardens. What we're sort of thinking at this point is that lower Attine agriculture might just prefer all the same microbes, or there might not be huge differences between the microbes that they prefer. And so that is something that we are looking into at the moment. You might be sick of hearing me talk about microbes, and you might be reminiscing on earlier parts of the talk where I showed you spiders and cockroaches and all sorts of fun things. And so you might be wondering, what sort of insects did you find in these gardens with your metabar coding approach? We didn't find any insect DNA in these gardens. So either there were just no insects in these particular gardens and we need to improve our sampling, or there were insects and we just didn't capture them with this approach. These are both sort of hypotheses that we are exploring more and using some positive controls in the future. How much time do I have? Okay, I'm just gonna go over one other really cool finding that came out of this work. And that has to do with Escovopsis, which if you'll recall was that specialized crop parasite. So we had 30 samples of fungus farming ant gardens in this study, and actually 10 of them were positive for Escovopsis. And this is weird because these were mostly healthy gardens, right? We didn't see any evidence of disease in any of these gardens. And so I mention this because it's adding to a sort of new and growing body of literature that suggests that Escovopsis is maybe not the disease-causing agent, but it might be just a microbe that takes over in stressed gardens. So not an agent of disease itself, but rather an opportunist in those sort of already distressed gardens as well. And so that just really kind of underscores the role that the ants and their actinobacteria are playing in structuring a healthy microbiome. And I would argue that it adds to this importance of evaluating these species interactions from a community context, because obviously if you take a species out and grow it in a sort of novel environment, it might be doing something differently than it's doing sort of in nature. So this was a long talk. Thank you all for sticking with me so late at night. Just gonna go over some key takeaways. Fungus farming ants are cool, that's obvious. There are many species that associate with them and they do really cool things, a little bit less obvious. Fungus farming ant gardens are good incubation chambers for vertebrate eggs. They're good for hitching a ride. They're great for studying microbial ecology from a whole suite of different aspects. And also really, really good for studying community ecology as well. So I just want to end on plugging this cool new resource that just came out literally yesterday, which is created by Research Quest and the Natural History Museum of Utah. You can actually go online and be a fungus farming ant and be a scientist. And more importantly, you can click on videos of Ted and Jeffrey and myself talking to, I think, third graders was the target audience. So I've linked the resource there if you're interested. And that is all I have for you today. So I want to take a minute to thank all of the people who helped with this work and made this work possible. Couldn't list them all, but these are just some of the people that I've worked with directly, as well as my funding sources and all of you. And thank you to ESW for having me. And I'm happy to answer any questions that you might have. Repeat it. Yeah, got it.
You refer to the leaves as substrate, but they could be a lot of different species of foliage and maybe flower petals and all sorts of stuff. What can you comment on that, or how does that help structure this?
Oh, absolutely. Yeah. So the question was, you know, I talk about leaves being a substrate, but these ants are feeding their fungi a bunch of different things like flower petals. They'll also feed their gardens insect frass. In some cases, they will feed their gardens decapitated heads of other insects. That's in the case of yeast agriculture. It's thought that they do that to promote that yeast-like growth. So actually, yeah, so the substrate that they are feeding their gardens plays a huge role in the microbial communities that are in the gardens, but also in the microbial communities that are in their guts. So if you recall the case of mollicutes, which are the nitrogen-fixing bacteria in the gut, there are different strains that will appear in leaf cutters that use grasses relative to leaf cutters that use like a shrubby forest plant. So yeah, we know that diet plays a really important role in structuring their microbiomes and sort of vice versa. Yeah, I'm glad you asked about diet because I cut that out of the talk because I didn't have time. So thank you. Yeah.
So there are carnivorous, like, or maybe not carnivorous, but they also are bringing like prey, right? Or maybe just invaders,
like eating,
throw on the pile.
Do you mean like the fungus farming ants eating prey?
Well, no, not the ants eating it, but they're feeding their gardens like other insects as well.
Oh yeah, yeah.
So
cool. Yeah, so the question was like, are they also feeding their gardens other insects? Yeah, yeah, they are. I know a lot less about what they're eating, but they kind of eat anything and everything. Insect frass. I'm told that sometimes they prefer like flower petals. I'm told that they're very picky sometimes, but yeah, I've never actually had to keep them alive myself. So you'll have to talk to Eugenia about that. Yeah, she'll feed them orange peel in the lab, or when we had them, she would feed them orange peel in the lab. So yeah.
South American map, Bolivia and Venezuela didn't have any mess.
Yeah.
They just, nobody was there.
Yeah, so the question was on the South American map, which maybe I can pull up. We didn't have samples from Bolivia and Venezuela, and sort of why. I'm not really sure why. I think at some point, you know, there were collecting trips that happened in Venezuela and Bolivia. I don't know if it was a geopolitical thing that led to that. We might have fungus gardens that are out on loan from those locations that I just didn't find. Ted might be able to tell you more about where we've been collecting.
I've never been to Venezuela. Nobody in the lab, I think, collected in Venezuela. Same with Bolivia.
Okay. So for those on Zoom, I guess nobody has been to Venezuela or Bolivia since Ted has been here. Should we go? Not right now, maybe. Sorry. Too soon. Yeah, we'll go to Bolivia. Yeah.
One question, chat. Jill?
Oh, okay. Can you hear me? Can you hear me?
Can I stop sharing, you think?
Oh, mute. It says mute. I'm unmuted. Hi, can you hear me?
All right, let's see.
I can do it.
Scrolling in the chat.
No, she's trying to talk. She's trying to say her question, but we cannot hear. We cannot hear.
Sorry, whoever is trying to talk, could you type your question? Because we don't have audio on you. Why do you use lower and higher agriculture? Are you referring to terminology or just in my research? Yes.
I think
Terminology.
so.
Oh, okay. That is just the terminology that I learned in terms of like agricultural systems. Maybe there's other terminology to use. So I would be glad to learn it, but that is just what I have always heard it referred to as.
Yeah, I think something like
I could see that. Yeah, it is like sort of a classist terminology. Is there other terminology that we can use?
Ask that.
People have criticized it, and it just basically kind of means ancestral and derived, you know. And lower is a paraphyletic group of ants, and higher is a monophyletic ant. So higher arises from within lower. I mean, it's just the terminology we've used for years and that I inherited.
Jill says we could perhaps use simple versus complex, few fungi versus many. Jill, I'm not sure if you heard it, but we just talked about derived or basal. So yeah, I think that's a good point about the terminology. So maybe we could have a discussion on that at some point. Yeah. Oops. All right.
I have a comment.
Okay, sure.
I would be very curious to see these tests on pellets, the pellets that the virtual queens take with them, just to see if there is horizontal.
Yeah, so it was a comment / question on the biodiversity of infrabuccal pellets. that has actually been done before. So people have been really interested in sort of the microbes in leafcutter ants. Do they come from the environment and then get filtered out, or do they come from sort of vertical transmission? And it seems like it's a mix of both. So some of the microbes come from the infrabuccal pellet, which is from the natal garden, and then some is from the environment. I don't know about lower attines, basal attines. Someone should do that.
Yes.
Yeah, I have no idea how you go about like collecting a new queen as she's coming out. I don't know how that would even work, but we could try.
Yes.
Yeah. Okay. I'm a question, Jesus. I have a question. It's been a long day. I'm a graduate student at UPenn, currently studying Aphesophiliotes with the goal of uncovering molecular links to behavior. The colonies are pretty sealed without room for other organisms to get in and out of. Do you think that limiting the ability of other organisms to interact in gardens will cause me to miss key behaviors? I'm not quite sure about the behavioral aspect. I have a feeling that behavior, I mean, it depends on what sort of type of behavior you're looking at, because certain microbes will elicit certain behaviors. Like if there's trichoderma in the garden, that will elicit the weeding behavior. But we are currently looking at, we had a bunch of colonies housed in the lab for a while, for many, many years. And so we're currently looking at how the garden microbiome shifts when you bring a colony from the lab into, or excuse me, from the wild into the lab. So I will look at that explicitly, and I'll let you know if there's anything alarming. But I think, you know, on the basis of behavior, you'd probably be okay. But I could be wrong. Please don't come after me if I am. Yeah.
You mentioned the frog laying eggs. What happens when the eggs hatch?
Yeah, so the question was about the frogs laying the eggs in the garden and what happens when the eggs hatch. All I was able to find on that was that the tadpoles are then cared for as ant brood. Yeah. I don't know if there's like more like water in there. That's all they said. So yeah. Yeah.
Back off of that one, where you find a lot of these sort of brood parasitites, are they near the top? Because I'm imagining right after you close, unless you're using a strategy like these frogs, you have to book it out of there. So I'm just curious, did they mostly put these brood chambers near the top and they have more successful with that?
You're talking about like with the parasitoids specifically?
Well, the wasp is one that stuck with
Yeah.
how they were pulling that wasp.
I know.
And I imagine they see a lot more success if they manage to stay near the mouth of the nest. I don't know.
Yeah, I mean, I think that intuition, oh, sorry. For folks on Zoom, the question was,
Just where do the brood chambers lie?
yeah, like where do the parasites tend to parasitize in these gardens? And is it closer to the mouth where they can get out faster? I think that intuition's probably correct, but also like the ants are growing new fungi. Like the fungus garden is going to shift in its architecture from the time that you laid the eggs. So I'm not sure if you have more success getting out if you are closer to the nest, but someone should look at that. Maybe a grad student somewhere. Yeah.
You know that some of the diapretids I've seen associated with aptites, they actually adopt a like tiny ant morphology. They're scaled way differently than their host ant colony obviously. They're shaped like ants, which is bizarre. I'm wondering if they're being ants-nated and instead of using chemical camouflage, they're actually just kind of attempting like, look, I'm just really small, leave me alone.
Yeah.
They escape.
Wow.
Diapretids are very bizarre.
Interesting. Yeah. So for folks on Zoom, it was noted that sometimes diapretids will pretend to be ants and disguise themselves that way. That's really cool. I've never seen or heard of that. The only thing that was noted in some of the stuff that I was reading about diapretids is that they very quickly will clean their antennae. I don't know why, but I guess they do that. Yeah.
So they're cultivating the fungi. Where does fungus come from? You bring nests,
Yeah. Yeah, so the question was, where do the cultivars come from in the first place? So usually it's vertical transmission. So new queens will grab a piece of the fungus in their mouths and hold them in their intrabuccal pockets, and then they'll disperse and use that to sort of seed the next fungus garden. But there are cases where they lose their fungus garden for whatever reason, and then they will steal fungus garden from like an adjacent fungus farming ant. In some cases, they might switch to a new fungal cultivar, although that's only happened once in the case of coral fungus agriculture. So it seems like maybe that's not a great strategy. It's only happened and worked once. But yeah, they are pretty opportunistic if they lose their fungus garden, but typically it is vertical transmission. That's a great question. Is there genetic variability in the fungus? I don't know if they reproduce sexually at all. Sometimes, so a lot of the lower attine fungus can grow in a free living form, so they don't need the ants to survive, and so they can produce mushrooms and stuff. But yeah, I can't say that I know much about the genetic diversity. Someone else who studies fungi probably does, but I don't. But yeah, it's a great question. Yeah. Are there any cases of more than one ant species cohabitating in fungus gardens? Yes, there are. So there are cases of hyperparasitism, where there is diaprid parasitoids that are they themselves parasitized by, I can't remember the name of it, starts with an O. So there's cases of hyperparasitism, and then there's also cases of multiple megalomyrmex species being in the same fungus garden as well. Yeah.
May I ask a question about the Corinth spider?
Oh, the Atocobius?
Yes.
Yeah.
Referred more than once to it's probably feeding on fungi, which strikes me as being very strange for a spider. How do we know that that's what it's doing? That's not preying on other symbionts, insects.
Yeah. Yeah. So the question was, how do we know that Atocobius spider is eating the fungus rather than preying on like the ant brood kind of thing?
Well, you mentioned it also affected the ant. Knew that it wasn't affecting other symbionts.
Yeah. So I don't actually know for sure what it's eating, and the papers that I read don't either. You're probably right. They're probably not eating the fungus. Spiders, I don't think like fungus. I don't know much about spiders. The short of it is we don't know what they're eating. It's just kind of presumed that they're eating something in the garden. But yeah, they totally could be eating stuff in the trash or other inverts. Yeah.
Would be really interesting. Fungivorous spider. I know there's one herbivorous one. So it's conceivable out there.
Yeah.
Very strange.
Yeah. I think people are sort of just starting to study Atocobius more. The papers that I found in those videos were from like the last 5 years. But I don't give much thought to spiders. Sorry for all the spider people. But yeah, it would be cool. Yeah.
Is the ecology of the chambers where they have these fungi in terms of floriculture homogeneous species? The question is, how destructive is your sampling?
Yeah. So the first question was sort of what does the nest agriculture look like across species? They vary quite a bit. So some species will just have their nests sort of open to the air, but under like a log or some dirt. Some sort of prefer being under roots, and then some you have to dig and dig and dig until you can get to them. So they do vary quite a bit. And the second question was, how destructive are your sampling methods? They're pretty destructive. For most of the sampling that we did in Pan, and these are not species that are like of concern, the ones that we were sampling anyway. I think if there were rare species, they would be a little more caretaken. But when we sample the fungus guarding, we do get the queen so that we can, study her and maybe keep her alive for a while. So the colony does not survive that. Yeah. Yeah. When it rains, do the chambers get flooded or are they built to thwart water entry? They do get flooded sometimes. We know that some gardens, I don't know this, Ted has told me this, some gardens will exist in areas that are completely underwater during the rainy season, and then the next season they're still there. So somehow they're surviving. I think there's been some literature on how they do this. And I don't want to say anything wrong, but if I'm remembering correctly, they do sort of some behaviors and moving their gardens to sort of try to stave out the flooding season. Like imagine if you're in sort of an air pocket. But yes, the short of it is they do exist in flooded zones. I don't know if they're specifically built to thwart water entry in other areas, but they seem like they're pretty hardy to water getting into the nests, which I was also surprised by. But yeah. Yeah. Thank you so much.
Thank you very much. We have a little thank you gift for you. It is in fact the first one.
Wow.
So yes.
Yes. No, just come to my.
I don't know if it's animal, but yeah.
Thank you. Yeah. Thank you so much. Yeah.
Okay. So thank you all for being here. So may I have a motion to adjourn our meeting? Second? All in favor? Thank you very much and have a good evening.